Hi, Oh, there we go. Hi, folks. Sorry about the poorly timed phone call. Yep, I'm at Drake University. I've been here about a year, formerly 20 years at the Iowa Department of Natural Resources, working on groundwater, surface water issues. And somewhere along the way, um, nope, I don't know how to shut this down. Sorry. Um, Somewhere along the way, I had a lot of questions about E. coli. We are working on manure and frozen ground legislation. Okay, my kid is having an emergency. It's always this time. Um, and um,
You want to hit pause, Claire?
yeah,
Okay.
give me like 2 seconds here because I just don't know
Yep, sounds good.
when she's repeating this thing. Hang on. Okay. I apologize, everybody. Anyway, so I went back to do my PhD because I was really curious about, what E. coli data really meant and what the risks are. With the manure and frozen ground legislation, we knew there was a lot of E. coli in some of the meltwater coming off of crop fields that had manure applied in the sort of those warm moments in March and April, but I really didn't understand. I didn't think anybody really understood what that meant for risks downstream during the recreation season. And so that's what really got me started. And along the way, I learned some genetic techniques which help us to understand where the poop was coming from. And so that would be the unofficial title of my talk today would be where's the poop coming from? A little less academic speak. Let's see. All right. So the work that I'm going to present to you today kind of comes from a lot of different collaborations. A lot of different folks have contributed time and effort. I don't have um, enough space on this slide to thank everybody. But this work has been started. Some of the stuff I did at DNR. We've used a lot of different labs, but the Lab for Infectious Diseases in the Environment up in Wisconsin, we've leaned on heavily. They're really experts in identifying whether there are pathogens in our water. And Cassie told me there's a lot of interest now in source tracking, and I'm going to show you some of the source tracking data that my students have been collecting. And we've been working more recently with Jonah Ventures for partners and some of that for the lab work. So I'll talk about that. Okay, so what we're really talking about is it safe to get in the water? And if we have a group of recreators here, I always want to highlight that the physical risks are the most serious in most cases, right? So first you have to be safe. So this is my slide to remember uh, to wear your life jacket. We will worry about getting sick next, but take care of your physical safety first if you're getting out in the water. Um, Waterborne diseases, we know they affect a lot of people, but the pathways, a lot of these same diseases that cause diarrhea, skin issues, that kind of thing, in recreators, they can be passed through recreational waters, through drinking waters, and also through food. I think one thing that's interesting about some of these statistics from the CDC is that, there's a lot of people get sick. A small portion of that are hospitalized, and the number of deaths, is a pretty small proportion of that. But if we look at food-related illnesses, same thing, sort of these microscopic organisms that are present in our water and our food, about the same number of hospitalizations. Quite a bit more foodborne illness out there, but similar as far as the proportion of hospitalizations. And these, if I go back, these waterborne diseases, this is nationwide data. They can come from a lot of different exposures. So this isn't just untreated recreational water. It's also pool water, drinking water, and other ways of contacting water, getting exposed. So what kind of organisms are we thinking about when we're thinking about recreational water safety? We talk about the ones that make us sick. Those are considered pathogens, and they come in a lot of different shapes and sizes. We think a lot about E. coli, which is a bacteria, but we can have large organisms like Giardia that form cysts, and really, really small microscopic viruses like norovirus, right? Really different sizes. They survive differently in the environment. And so, I really want folks to broaden their mind a little bit beyond E. coli, because I think we're talking about a lot of different organisms that might be out there. For the purposes of this talk, I'm talking only about viruses and bacteria and protozoa. I'm not going to talk about the blue-green algae, which is a whole different system, whole different ballgame. But we do some of that work. Recently, as part of the Central Iowa Source Water Research Assessment, we were able to gather some public health data from Polk County. And this is numbers of infectious diseases that are reported from the county from 2019 to 2023. So these are some of the organisms and illnesses that folks in Polk County, are experiencing. They're not necessarily recreation related. They can be, right? But these are the organisms that are out there: Salmonella, Campylobacter, Cryptosporidium, E. coli, Giardia, right? So those are some of the big ones. And it's always interesting to look at these organisms over time or seasonally. When we have recreational exposures, those tend to be the highest in the summer. So if we can look at infectious disease data over time, we can sometimes tell whether there's more likely to, it's coming from recreational exposures. So one example of those data, I think there's kind of interesting, was Campylobacter. So you can see both in Polk County and Sac County, some seasonal trends, right? So that means that there's more transmission during the summer, which could be from swimming. It could be from warm potato salad at picnics. It could be a number of different things that are seasonal. But if we see that seasonal signature, it at least means it potentially could relate to recreation. There's this one spike in Sac County in August, and I don't know if that's a county fair where a bunch of people were hanging out with some animal that wasn't so clean, or they just didn't have the immunity and they all went to the fair and got sick. I'm not sure what happened there. But that's, the kind of thing we're looking at when we're trying to understand diseases and recreational exposures. What we are most familiar with, I would say, are the indicator organisms, right? I have this little circle diagram here. We have a huge family of bacteria that like to live in warm, organic, rich places like our guts. We call those the total coliform. Within those are the ones that specifically live in the guts of warm-blooded creatures. Those are fecal coliform. And then a subset of those are the E. coli. and a much, much smaller, not these not to scale here, subset of E. coli are E. coli that can actually make you sick, or the pathogenic E. coli. So we have to be careful when we talk about E. coli that those are indicators. They don't necessarily make us sick, and only a small subset of the E. coli are really pathogenic. But the presence of E. coli was studied along with epidemiologic data, health data, places like the Great Lakes, the beaches in Chicago, where a lot of people were recreating. And there is a correlation between E. coli and how many people get sick, or total coliform and how many people get sick. And based on those correlations, the water quality standards that we have are set at the level of 36 illnesses per 1,000 contact events or recreation events. That means that if you swim in water at the E. coli standards, the current E. coli standards, that's your estimated risk, right? 36 illnesses per 1,000 swimming events. I'm not sure exactly where they came up with that number as far as acceptable levels of illness, but that's the number, and they've stuck with it. I like to modify this graphic a little bit, just to say that we're finding that there are E. coli that don't need to be in the guts of warm-blooded animals. So we have more questions about E. coli and whether they're really good indicators of risk, because if E. coli are growing in our environment, in our beaches, for example, and they're not related to recent inputs of fecal material somewhere, then they're not necessarily telling us what we think they are as far as risk. Right. Okay, so you folks are probably pretty familiar with the standards for E. coli in recreational waters, right? We have a geometric mean, just the average of samples over a period of time, and then the single sample maximum. So the magic number for both beaches and streams is the same, right? If you're above 235 organisms or MPN per 100 mil, then you're above the standard. And that's for primary contact recreation, right? So that's swimming, getting your head underwater, things like that. If you're kayaking, splashing around, but not likely to be accidentally ingesting a whole lot of water, then the secondary standards apply. These a little lower. So that's the 630 for the geometric mean and the 2880 for the maximum concentration. But again, these are the same for beaches, any water body, lakes and rivers, right? And the smart folks at IHR have looked at all the E. coli data statewide, and it's pretty clear that rivers always have higher concentrations of E. coli than beaches, which have higher concentrations than the open lake water, right? I mean, if you take all of the data statewide, that's the pattern, right? And we're still applying the same recreational water quality standard in rivers that we are at beaches, but the systems are really different. And when we have streams, we can suspend a lot more sediment. We're moving stuff from up higher in the watershed. We just always have a lot more E. coli. And this leads to our impairments, right? So beaches, E. coli or indicator bacteria are sort of the fourth leading cause of impairment at lakes, and they're the primary leading cause of impairment in river systems, right? And so this makes us think, gosh, there's a lot of poop in the water. What are we going to do? But in order to figure out what to do about it, we had to figure out who it's coming from, where it's coming from. We also want to make sure we're not spending a whole lot of money to clean up E. coli if it's not associated with risk. So we really have to do a better job figuring out if E. coli are good indicators of risk. And then to understand risk, we need to understand what type of activities or exposures are happening, when and where those risks are highest. For example, flow matters quite a bit. So understanding how risk relates to flow, and then, of course, what we can do about it all. So in my lab here at Drake now, we use sort of the traditional let's measure E. coli methods, but we're pairing that with these more advanced genetic methods, right? So with the genetic methods like qPCR, which is what we all had when we had COVID, right? We had a qPCR test. It's a unique genetic sequence that's associated with a particular organism. And so if we count those genetic sequences, we can get an estimate of the quantity of whatever organism we're looking for in the water. And we use the same type of method to find specific pathogens, but also to identify the sources of fecal material in the water. So we can look for sequences that are specific to bacteria that are in the gut of animals and humans. And so that's how we're using those methods and kind of putting them together to better understand our systems. Okay. So yeah. So I'm going to split the rest of my talk into beaches versus streams, because as I've said, they're really different systems, and what we understand about them is really, really different. So most folks on this call are probably familiar with the DNR's ambient beach monitoring program. You know, they go out and they sample 9 locations in the beach area. They composite that, and they use that to determine whether to post a warning, right? So they do the sampling early in the week, they get the results, and they post the results for the weekend. We all know that E. coli levels can vary pretty significantly over a couple of days, but it's the best system we have. What Jason Palmer at DNR has been working on for several summers now is looking at the relationship between E. coli in the sand and E. coli in the water, because he found that the E. coli at the beaches were really high and the open lake water didn't have E. coli, and there was no correlation between what happened upstream in the watershed of a lot of these lake systems and what was happening at the beach. So he decided, you know, we really need to focus on the beach environment itself, close to that water. He found these blue bars here are the water samples. He always found high concentrations of E. coli in the shallowest water, and then lower concentrations as you got further away from the shore. And he's like, oh, well, then, you know, the bacteria are coming from the beach itself. We looked at concentrations of E. coli in the beach sand, and sure enough, they're quite high, right? So for the same little square centimeter of sand versus, this equivalent amount of water, much, much higher concentrations of E. coli in the beach sand. So his, hypothesis is that a lot of the E. coli we're seeing at beaches is generated right in that beach system itself. And it can be transported in a bunch of different ways, right? It might be a rainfall, it might be people mixing that, it might be wave action. There's a number of different ways. And so if you look at parameters like rainfall versus E. coli, they don't correlate as well as we hope because there's so much complexity in those beach systems. So what we did following, Jason's initial studies was to start looking for the pathogens themselves. And we did a whole long list of different pathogens at a couple of beaches, 2016 and 2017. And out of our big long list, we only found 4 of these different organisms, and only 2 of those are infectious to humans, right? So we found adenovirus and noroviruses; those are infectious to people, but we also found bird flu and this plant virus, the pepper mild mottle virus. That's a mouthful. But pepper mild mottle virus shows up in all of our wastewater. So it's most likely coming from like all of the pepper products that we use, like cayenne pepper, our salsas, that kind of thing. So it's just this little bit of moldy pepper that's in human wastewater. But so we did this big long list, and we really only found a couple. And if we look at where we found those organisms, most of them were at McIntosh Woods Beach on Clear Lake, 2016. 5 different samples that had hits. Hickory Grove had a couple samples with the adenovirus. But for Polk County folks, the Bay Creek State Park, we sampled 13 times in the summer of 2016, every other week for the full summer, and we didn't get a single hit of a pathogen. And the process that we're using is to pump water through a dialysis filter for an hour. So you get, 100 gallons was sort of our goal most of the time. So you're filtering a lot of water. So the fact that we didn't get hits isn't because we didn't sample enough water. It wasn't there. And their detection limits for this lab up in Wisconsin, are on the order of 2 or 3 gene copies per liter. So we don't have to have much genetic material to get a hit. Jason went out again in 2017, a couple of different systems, but the story was pretty similar, not a lot of pathogens. And then we decided to venture into source tracking. So our first adventure was with a fee-for-service lab called Source Molecular in Florida. And this is not a scientific study as far as like representing a lot of different seasons and flow regimes and everything. It was just times when we thought there might be something in there and it was worth spending the money. And so you will see from that initial set of source tracking a number of different things. We see that you can sample twice and get completely different results, right? So this McIntosh Woods Beach sample had bird, goose, and dog hits in one sample and then nothing in another sample. At Hickory Grove Beach, the goose hits were pretty consistent. A little bit of bird and dog in those samples. And then Beeds Lake, poor Beeds Lake, pretty much had everything in it. Kind of a slimy, disgusting mess. But oops, sorry. But there's a lot of talk about CAFOs and how swine are the cause of all bacteria-related problems in Iowa. And that may be true for Beeds Lake, but it's not. We haven't seen evidence to support that in these other beach systems. Okay. So that's stuff that happened at DNR. And we've been looking around at some local systems. We're using a lab called Jonah Ventures out in California, and they will do a suite of targets. I added a few extras to these, but they will normally for $135 you can find. I know I'm schlepping for them. They should give me royalties or something, but they ship you this nice little sampling kit. Oops, it's not focused. There we go. And it has a syringe and a filter and a DNA preservative and some gloves. And you go out and fill up the syringe, pass it through the filter, and ship it without any ice, or overnight shipping fees. And so they get good results and it's really easy to use. So I highly recommend if you're wondering whose poop is in your water, running these kits. What we have found has been really interesting, right? So this figure is sort of related to Gray's Lake and the tributaries going into Gray's Lake. The beach itself had human hits both times that we've sampled, once in the fall of 23 and once in the spring of 24, and a teeny bit of dog DNA. The discharge to Gray's Lake, there's really sort of one culvert that goes in. We had a mystery. We have a whole bunch of swine specific genes in the middle of town. Okay, so there's no confinements between the golf course and Gray's Lake, but we're still getting some swine hits, and bovine and poultry as well. So I don't know what the answer to this one is. Maybe there's a pet pig in the watershed, or maybe there's a leaky sewer and we shed quite a bit of swine-specific DNA in our human waste from what we eat. It's also a possibility. Anyway, and then we had a lot of raccoon DNA in the fall, and even a larger percentage in the spring. But those it's pretty clear that what goes into Gray's Lake at one end and what's happening at the beach are really separate. So we could put a whole lot of effort into, stopping whatever waste leaks are happening and going into Gray's Lake. But it seems like, as far from the practical point of view, wherever the human waste hits that are showing up at the beach probably be the priority as far as cleaning up any potential risk at Gray's Lake. Looking at time. Okay. So the understanding lakes and beaches is, one thing, and then I hinted at some stream data. So then the second part of my talk is looking at stream and river systems. One project that we did, sort of pre-Icon water trails, right, with working with Polk County Conservation to understand what the risks are in some of our local waterways that are going to be used a lot for recreation. And so you may have seen these boxes with the help of the folks at IHR. We installed some ISCOs, and those sampled every 2 weeks. And then the samples were sent to Wisconsin for pathogen and source tracking analysis. There's the ISCO. There's my water monitoring supermodel child. And similar to the beach work that we did with DNR, there's a whole long list of things that could make you sick that we tested for. The results are pretty interesting of all those samples. As far as protozoa, the most common was Cryptosporidium. For bacteria, it was Campylobacter, which, if you happen to remember way back to the Polk County health data, shows up pretty frequently in Polk County. And then the highest concentration of a virus is rotavirus. We left that one off of any risk assessment because we now get vaccinated or vaccinate our kids for rotavirus, and they can shed some of the virus without it necessarily being infectious. So just because it's there doesn't mean we're necessarily going to get sick from it. We'd have to look more closely to understand. But the big overarching analysis based on the number of pathogens in these waterways is that if you look specifically at the pathogens and how much exposure folks have, Poor Beaver Creek had the highest risk. Walnut Creek came in second, Raccoon River, and then the two different sites at 4 Mile and the Des Moines had the lowest risk. And the different colors are different types of contact. So we have blue for swimming of all ages, and then orange is for swimming children 6 to 12. So the fact that that bar is higher isn't because children are more susceptible to the illness. It's just because they tend to drink more water accidentally when they're swimming. And then, limited contact, the kind of kayaking, splashing around, things like that. With head immersion, I don't know if you fall in with your kayak is the green, and then purple is the limited contact with no head immersion, right? So this data set is from 2020 and 2021, relatively low flow conditions, right? We all remember it was pretty hard to recreate on the streams during those 2 years. We also know that there's been quite a bit of wastewater improvement in the Beaver Creek watershed. I think I see Evo on the call, but others, like there's improvements at Camp Dodge. There's shut down the Grimes wastewater treatment since this study. So a lot of stuff is happening. And we're really hoping to go back next year and see the risk at Beaver Creek go down. And what you're seeing on this plot is also average risk over 2 years, right? It's not telling you at high flow or low flow what your risk is. This is the average. So it could be quite a bit higher, I think up to 200 cases per 1,000 recreators on the worst day, or quite a bit lower, right? These are the average risk values. Beaver Creek is fascinating because if you look more closely and you look at discharge, almost all of our samples were at really low flow. And that's when the pathogens showed up, which is pretty clearly a wastewater problem, right? When you have a lot of bacteria and viruses at low flow, it's not likely that they're being washed in from up higher in the watershed as much as they're coming from a continuous discharge of some sort. And if we look at the source tracking that went along with it, the Beaver Creek definitely had a mixture of sources, ruminants, pigs, humans, and birds, and cows. There's a little overlap between cow and ruminant. But either way, you can see for Beaver Creek, there's quite a bit of human indicators showing up, and at really high concentrations. And that's in contrast to the Des Moines River that also had some human indicators, but at much lower concentrations. Again, for folks who are interested in the role of pig poop in our water, the little purple dots are pig indicators. And they did show up in October and November samples. One in an August sample in the Des Moines, but they certainly weren't there all year round. All right, I'm getting close to the end here. So we have the DNR study at beaches. We have Polk County study 2020, 2021 in streams nearby. And then since then, my students have gone out, to practice the techniques. And this is Cassie helped us with the spring snapshot that Polk County does. And again, we're looking at both E. coli and the source tracking. These data are from the Raccoon and Des Moines River watersheds. This is pretty typical of what we get from the Jonah Ventures lab. So green is human sources. We do find a lot of human waste indicators showing up, especially in our urban areas. I would be very careful not to take the height of these columns, put too much weight on that. The gray and black dots and line are E. coli concentrations. And so sometimes you get a lot of genetic material, but you have low E. coli concentrations. And sometimes you have low amounts of genetic material. I don't have a good example of that, but in high concentrations of genes. So don't put too much weight on that. But I would say the take home from these data are there's a fair amount of human waste out there. We do see pigs and cows and poultry and dogs showing up in these systems. And if we go to the same site repeatedly, the distributions do change. So it's not just a mixture of sources, but it's a mixture of sources and different timing. This figure, just to highlight sort of the timing issue, was 3 samples that we took during an event at Beaver Creek. So in the spring, in April, we had an ISCO running at Beaver Creek. We took a sample before a storm event, kind of at peak flow during the storm event, and then during the receding lambs, as the water levels were going down. And the gray in these bars is raccoon specific genetic targets. And so we saw a lot of the raccoon influencing sort of the early flush from this rainfall event, and then pretty much absent in the last sample, which tells me, and I guess I would want to repeat, this more than once before I drew a really hard and fast conclusion, but that there was a fair amount of raccoon poop in sort of the stormwater infrastructure, the drainage, that washed out at the beginning of the event, and then wasn't as big an issue in the last sample. You'll see, like this is the extended list of targets. So geese and white-tailed deer were also run. So that's just, timing matters. Time of year, timing relative to a precipitation event. So overall takeaways, there's always risk. If you're communicating to people about recreation in Iowa, just tell them there's always risk, and we can never tell you exactly what it is. So you should always take precautions. Wash your hands, try to minimize ingestion of water. At beaches, that sand, nearby environment's important. River systems are always going to have higher E. coli, but we're really trying to understand if that also means there's higher risk. And while we do get higher E. coli, tends to go up with high flows. Some of the highest risk is actually at low flows, where we have human wastewater discharge dominating. We have lots of different sources in our watersheds. E. coli are a good indicator, but not perfect. And if you go out and do source tracking, you're going to find some weird things. So lots more questions. And with that, I will just highlight that I am a recreator in Iowa streams, and I take my kid and my dogs to Beaver Creek, despite what I know about the level of risk. So I do think it's important to get out and to understand that there's risks out there, but also just there's a lot of benefits to being on our waterways. Cassie, you want to let me know if folks have questions?
Great. Yep. Thanks, Claire. That was a really interesting presentation. If anyone has any questions, please go ahead and type them in the chat now. We don't have any at the moment, but we will wait about a minute to see if anything pops up.
If you guys don't ask questions, I'm going to start asking you questions. Hey, one of my students is on the call. All right. Eva, when's Camp Dodge going to be done? He's not going to.
I don't have the answer to that one. Sorry, I was on mute. But since you got me here, let me ask, can you expand on the variability of pathogens? Because it seems that sampling, like you said, timing and spatial variability can be very significant. And sometimes we can try to make decisions based out of a few samples that really don't give the big picture.
Absolutely. Well, what I can tell you is, when we sample at low flows or during, drought periods, the human waste, signature, it definitely dominates. But we still had mixtures, and the risks, the actual presence of pathogens occurred when we had mixtures of different sources. So the academic paper that came out of all that Polk County work really highlighted that you couldn't tie any individual pathogen to a particular source, right? So we have really messy systems with lots of different sources. And then, we're really hoping to do similar work at higher flows. Would have been nice to do it this year. But, the source tracking we've done this year, we're still seeing a lot of the human waste at the high flows as well. So it hasn't gone away completely and switched to, up higher in the watershed sources. But you have to have a minimum of, I would say, 24 samples to characterize risk in a given system, and then to be able to break that down further to help, tease out flow or, other parameters would be even more. So it does take a fair amount of work to get a really good idea of what's going on, what the risk and the variability is in a system. And even with 24 samples, that Polk County study was very much dry. So we do need a little bit more sampling before we can give a strong characterization. And even when we do that, there's going to have been change, right? These wastewater systems in the Beaver Creek watershed, some of them are going to be completely cleaned up. And so we will see, differences over time. So very dynamic systems.
Great, thanks. We do have a question. Could you please review again the E. coli standards for various types of contact? I'm especially interested in children and swimming.
Right. Let me go. Don't get dizzy. I'm going to flash backwards here. There's an easier way, but I'd have to mess with this side. Oh, and then I missed it. Okay. Right. So the E. coli standards, again, they were based on studies and people who were getting sick. None of those studies happened in Iowa. But the magic number, sort of the maximum concentration for E. coli in a system that tells you you're at that 36 out of 1,000 recreational events for primary contact. Class A is primary contact, which means or children's recreation. That means you're getting your head underwater and you're likely to ingest something, right? That's the 235. It's almost, it's an order of magnitude higher concentrations if you're not getting your head underwater, right? Because now you're not ingesting much water. And so that magic number is 2880. And again, it's organisms per 100 mil or MPN per 100 mil. So the concentration has to be a lot higher if the kinds of recreation you're doing don't involve much ingestion of water. But we know kids, right? They tend to drink, accidentally drink or on purpose, even when I'm yelling at them, like a fair amount of water. So the sample maximum is, you could take an individual sample and get a result, and you're higher or lower. The geometric mean they use, especially for the beaches, to say, wow, the concentration is on average a lot higher all the time, right? So that's a better indication of the conditions in general over a longer period of time. And so if you're always exceeding that geometric mean standard, then, you kind of have a problem lake or a problem beach. And so they can get on the impaired waters list either way, by exceeding the single sample max or by exceeding the geometric mean. So in fact, they get on both ways at this point.
Right, another question. What are your thoughts on health risks regarding goose poop sources? For example, small ponds with trails around them.
So in general, the human waste is most likely to contain pathogens that impact humans, right? So we're spreading things that, cause us infections. But birds, cows, pigs, there's a number of organisms that can be in the fecal material of any organism that can infect us, right? So usually if it's goose poop, it's gross for sure, but the risk is a little bit lower than if it was human waste sitting there in little piles on the beach, right? But there's still risk because there are organisms like Campylobacter crypto that can infect either animal. No, but in general, the human waste, just because it's the same organism, those pathogens are more likely to cause problem.
Right. Any other questions for Claire? Right. Seeing none, I will once again thank you so much, Claire, for this presentation and for being our speaker this month. And thanks everyone for tuning in today. August 13th is our next Lunch and Learn, and I'll be sending out an invitation for that shortly. It's going to be an update on the Icon water trails. And if you're not currently on the email list to receive information about these Lunch and Learns or any other business with the Central Iowa WMAs, please feel free to send me an email and I will put you on that mailing list. So have a great day, everyone. Thank you.
Thank you.